Hello everybody, welcome back. Today we're going to be going over chapter 13, Glycogen Metabolism, Glycogenesis, and Glycogenolysis. So the objectives, first we're going to describe the structure of glycogen. Remember, glycogen is the storage form of glucose. We're going to identify the primary sites of glycogen storage in the body and the function of these deposits in these tissues. We're also going to outline the metabolic pathways for the synthesis and degradation of glycogen. So synthesis, glycogenesis, degradation, glycogenolysis. We will describe the mechanism by which glycogen is mobilized in the liver in response to glucagons and in muscle during exercise and in response to epinephrine. A little more hormones and stuff that we'll talk about in relation to glycogen and its storage versus release. So, explain some of the glycogen storage diseases in muscle and liver, and describe the counterregulation of glycogenolysis and glycogenesis. So, glycogen is the polymer of D-glucose. Remember, we talk about polymer. Take a minute to remember what that is. Okay. It is a storage form of a polysaccharide present in tissues of animals, including humans. The glycogen synthesis occurs in all tissues. However, the liver synthesizes most of it, and then the muscle tissue. The liver synthesizes most of it, and then the muscle tissue. Glycogen is stored as large cytosolic granules. The major function of liver glycogen is to supply glucose to other tissues via blood circulation. Please remember this. This is probably one of the most important parts of this whole thing. Understanding that the liver glycogen is what we're going to be talking about a lot when it comes to maintaining the proper blood glucose levels. This is blood circulation. So it does via blood circulation whenever there's a need for energy production by those tissues. For example, in the fasting or starvation conditions. Fasting and starvation conditions typically starts, give or take, after 8 hours of not having any food. Liver glycogen can be depleted within 12 to 24 hours of fasting or starvation. And the function of muscle glycogen is to release glucose and oxidize it to supply energy for muscle contraction only when the muscle tissue glucose are depleted. Remember, so once glucose enters a muscle cell, it cannot leave. So the glycogen stored inside of the muscle cells stays inside of the muscle cells. Muscle tissue does not supply glucose to other tissues for oxidation. It only uses within the muscle for itself. Glycogen is highly branched and contains both linear chain with alpha-1,4 linkage and branch chain with alpha-1,6 linkage. The alpha in this case is specifically referring to kind of the structure of where the oxygen and OH, or the hydrogen and the OH groups are within the linkage. Just know, always look for the alpha. That's what we're going to be dealing with. We're going to be dealing with alpha. We won't really be talking about beta at all. So remember to keep that in mind, and when you look for answer choices, look for the alpha. 1,4 linkage, associate 1,4 linkage with linear, and the branch chain with 1,6 linkage. So what this is saying is the 1,6 and 1,4 are talking about the numbering of the carbons. So the 1 carbon on one glucose connects to the 4 carbon on another glucose to form a linear chain, and the 1 carbon forms a linkage with the 6 carbon on another glucose to form a branched chain linkage. Branching helps this molecule to fit within the cell, to be solubilized. That's a weird word. It's in order for it to be soluble, in order to be present there, and really able to release glucose units during cellular demand for oxygen and yield energy. Structurally and chemically, a glycogen molecule containing n number of branches has n plus 1 numbers of non-reducing ends. However, a glycogen molecule only has one reducing end. Excess glucose is converted to polymeric forms for storage as glycogen. If you see polymeric, just understand that the definition, that just means polymeric means more than one, poly more. So it's multiple molecules of glycogen. So here we have our individual glucoses, and we can say this form of linkage where the H's are both on one side and you have the O. This is what it's referring to as that alpha linkage. That structure is the alpha part of it. Beta, it's a little bit different where you have an OH and an OH on opposite sides. It's just a little bit different. We don't have to worry too much. We don't have to worry about that right now. So we can see this linear chain here all the way across. And if we number our carbons, this is our carbon 1, 2, 3, 4, 5, and 6. The 1 carbon connects to the next carbon's 6 carbon. 1 to 6, 1 to 6, 1 to 6. Sorry, I apologize. 1, 2, 3, 4, 1 to 4. 1 to 4, 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 1 to 4. And that creates our linear chains. The alpha 1,4 linkage. Now, the alpha 1,6 linkage right here, our 1 carbon to our 6 carbon, you can almost think of it, it almost kind of creates this like step down. So it's connected here, connected here, connected here, and there's like a step down here so they can connect to more, and a step down, and a step down, and a step down, etc. Just so that you can fit a whole bunch more carbons all together, or sorry, a whole bunch more glucose all together in a smaller spot, because you're taking full advantage of all of the area rather than just being a straight line. And we'll see more of that a little bit later. So the liver and muscle tissues are 2 major tissues that store glycogen. In the muscle, the need for ATP triggers conversion of glucose to glucose 6-phosphate that enters glycolysis. In the liver, the low blood glucose triggers glycogen breakdown to G6P, and G6P is hydrolyzed to glucose. Glucose is released into the blood and delivered to the tissues. So this is, remember how when we bring glucose directly into the tissue, so into muscle tissue, the hexokinase turns it into the glucose 6-phosphate. So in muscle, when we need ATP, we break down the glycogen to get it to glucose 6-phosphate, and then it enters glycolysis. In the liver, we break down the glycogen to G6P, and we remove that phosphate off of it, and then it can actually leave the liver and go back into the tissues, or go into the blood to be delivered to other tissues that need the energy. So the removal of glucose from glycogen. So glucose residues, that's what we're going to call the single glucose, the glucose that are getting stuck together, each individual glucose is a residue, are removed from the non-reducing end of a glycogen chain. It continues to remove glucose units until it reaches the 4th glucose unit from a branch point. Glycogen breakdown is catalyzed by glycogen phosphorylase. And we'll get more into all of like the actual images and how this happens later on. This is just the setup, kind of what we're talking about. With glycogen breakdown, there are 3 enzymes that are involved: glycogen phosphorylase, glycogen debranching enzyme, and phosphoglucomutase. So first one, glycogen phosphorylase. So the glycogen does glycogen phosphorylysis, which is bond cleavage. It's going to be breaking bonds. So glycogen plus a phosphorus, we get glycogen N-1 residues. So this N-1 is saying that we have released 1 glucose off of it, which gives us a glucose 1-phosphate. And it uses the pyridoxal phosphate as its coenzyme. So our non-reducing end and our reducing end. We think our reducing end typically would be the end that we would continue to be adding glycogen to. So we're going to be taking from the non-reducing end. So right here, this enzyme comes in. It's going to cleave that bond, replace it with a phosphorus, and we end up with glucose 1-phosphate. And now we have the next glucose is the non-reducing end, and that is our glucose N-1, N being the total number of glucose residues in that chain of glycogen. Not sure why that's popping up. Go away. Here we go. So that is one step that is occurring. Now we have glycogen debranching enzyme. Because remember, we don't just have linear chains of our glycogen. We also have branches that go from the 1 carbon to the 6 carbon. So the glycogen debranching enzyme contains 2 enzyme activities. Debranching, or glycosidase activity, catalyzes the removal of glucose units one by one from glycogen 1,4 linkage as glucose 1-phosphate. And 2, the transferase activity transfers the last 3 glucose residues from the 1,6 branch point, leaving the 4th glucose residue bound to the 1,6 linkage. Next, the debranching activity removes the 4th glucose unit from the branch point as free glucose. So what this is saying, our non-reducing end, so we have our glycogen phosphorylase is chop, 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 chop on this end until we get to the last 4. Now at the last 4, it's the glycogen phosphorylase breaking all those off. At the last 4, we have the transferase activity of the debranching enzyme. So it's going to take these 3 and move them onto the longer chain, just like that. It's going to transfer them to the longer chain. See how they're already pretty close? It's going to just pop them over and transfer them over there. And that leaves us with 1. And the glucosidase activity of the debranching enzyme, so that debranching enzyme is then going to pop off that last, it's going to break that bond and pop off that last glucose, essentially ending that chain, or that branch chain. And now our glycogen phosphorylase can come back and continue in its happy way all the way through breaking these bonds until it reaches another point where there's a branch chain, and it will do the exact same thing. Hopefully that all makes sense right now. If not, reread through this, re-look at this chart, and understand kind of that step by step what is occurring and what the function of each of these enzymes is. Now, phosphoglucomutase. Its job is it converts the glucose 1-phosphate to glucose 6-phosphate. Remember, so the glucose 6-phosphate is typically what we put into glycolysis, as well as what we then need to dephosphorylate in order to have it leave the liver. So the G6-phosphate can enter into the glycolytic pathway to produce ATP in the muscle, or it can be converted back to regular plain glucose in the liver to be sent up to the rest of the body. So this reaction catalyzed by phosphoglucomutase. The mutase is the key what we're looking here, where it's just mutating, it's changing the structure of the molecule. So in this reaction conversion of G1P to G6P, this reaction is catalyzed by phosphoglucomutase. This enzyme contains a serine hydroxyl group as it's at its active substrate binding site. This seryl hydroxyl group is phosphorylated in its active form. Now, during the catalyzed reaction, the seryl phosphate from the phosphoglucomutase is transferred to the 6 carbon of the G1P, forming glucose 1,6-bisphosphate intermediate, and the enzyme phosphoglucomutase is converted to its dephosphorylated inactive form. In the next step, the phosphate group from the first carbon of the glucose 1,6-bisphosphate is transferred to regenerate the active phosphorylated form of the phosphoglucomutase, yielding G6P. So hopefully that made sense, where essentially what we have. This is our phosphoglucomutase. It's going to come in here, attach its phosphate to the 6, and then come back over here and pull off the phosphorus, or the phosphate that is bound to the 1 carbon. That's its process. So it sticks on another one where it's supposed to be, and then pulls off the old one to leave us glucose 6-phosphate. So it kind of trades the phosphates with the glucose 1-phosphate. Now, hydrolysis of glucose 6-phosphate by glucose 6-phosphatase. So our G6P, remember we have to get it back to regular old glucose. This is specifically we're talking about in the liver, because remember the muscle, it's not going to be leaving the muscle tissue at all. But in the liver, G6P produced in the cytosol of the hepatocytes, liver, can be transported to other tissues via blood circulation. G6P is an ionized or phosphorylated molecule, and thus membrane is impermeable to this and cannot cross the plasma membrane. However, endoplasmic reticulum membrane has a special transported T1 system for G6P. As a result, it can enter the endoplasmic reticulum lumen, where G6P can be converted to glucose by the catalytic action of ER membrane-bound glucose 6-phosphatase. So remember, big key thing to remember, that in the endoplasmic reticulum of the hepatocytes is where G6P can be converted to glucose by the glucose 6-phosphatase. Now, glucose is transported out from the ER lumen to the cytosol by specific glucose transporters, localized in the ER membrane, and the cytosolic glucose is transported to the circulation by hepatocyte-specific glucose transporter GLUT2. Pretty standard procedure there. Okay, now talking about the regulation of the glycogen phosphorylase. So there are two ways of regulation. So we have allosteric regulation, which the inhibitors of that, meaning our ATP, glucose 6-phosphate, or glucose, if any of these are present in large quantities, it's going to inhibit the glycogen phosphorylase, which is going to be whose goal is to release more glucose. An activator is if we have lots of AMP, meaning that our ATP has been depleted. And this is our allosteric regulation. Now, covalent modification, the phosphorylated form of phosphorylase A is active, and the dephosphorylated form, or phosphorylase B, is inactive. So you can literally have phosphorylation or dephosphorylation. Think about probably most likely by a kinase enzyme of some sort, is going to activate or deactivate our phosphorylase enzyme. And here's a whole paragraph all about that. So let's dive in. The phosphorylase enzyme exists in two forms. One is its inactive form, which is dephosphorylated, and the other one is active phosphorylated form. Phosphorylation of its serine hydroxyl groups by phosphorylase B kinase, just like I said, most likely a kinase, activates this enzyme. So phosphorylase B is the inactive form, phosphorylase A is the active form. It activates this enzyme, and this form is termed as phosphorylase A, or active. Phosphorylase A enzyme requires pyridoxal phosphate as its coenzyme. Another enzyme phosphorylase A phosphatase, there we go, removes the phosphate group attached to its serine hydroxyl residue. The dephosphorylated form is inactive and is termed as phosphorylase B. Phosphorylase A kinase activity is stimulated by glucagon and catecholamines. Remember glucagon versus insulin, all its functions, and then the catecholamines such as dopamine, norepinephrine, and epinephrine. Remember, epinephrine is referred to as our adrenaline, norepinephrine is our noradrenaline. And also by increase of cytosolic calcium ion concentration, indicating a dependency on calcium ion induced pathways, such as via calcium calmodulin dependent pathways. You'll learn about these pathways next trimester in Physiology 2, talking about the muscle and how signals propagate through and how they use calcium in both smooth and skeletal muscle. We also have increased concentration of cytosolic AMP, which may be elevated during increased ATP utilization or increased ATP breakdown. Both the axogens of glucagon and epinephrine are mediated through specific G protein coupled receptor systems. Note here in the muscle tissue during skeletal muscle contraction, normal and during exercise, energy in the form of ATP is required. Therefore, generating the phosphorylated form of this enzyme is essential. That would lead to release of glucose from glycogen, followed by its oxidation, producing energy. AMP could be a source of ATP by the action of nucleotide monophosphokinase. Next, think about the role of glucagon. Glucagon is released in response to low blood glucose levels. This signals that there is an energy need within tissues. So it is obvious that this hormone plays major roles too in activating the phosphorylase A kinase to enhance glycogen breakdown, followed by glucose oxidation to produce energy. There's a lot of words in here. Big things I want you to focus on. Understand the phosphorylase and the B versus the A, the phosphorylase A and the phosphorylase B, what those are, which is the active, which is the inactive. Understand what activates this whole system. So the glucagon, the catecholamines, high calcium concentration, and also increased concentration of that cytosolic AMP, which again exactly what we were looking up in here. And really that's the most important stuff that I just talked about. Okay, mechanisms of action of glycogen phosphorylase by covalent modification. We have phosphorylation and dephosphorylation. So the catecholamines and glucagon mediate their action via activation of the specific G protein coupled receptors, activating that G subunit with subsequent activation of adenylyl cyclase enzyme to produce CAMP as the second messenger molecule. Amplify the signals multiple times to further activate protein kinase A and relate substrate protein phosphorylation, leading to a final action. That should all sound very, 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 very familiar. All of this here was our G protein coupled reactions that we studied. I think it was all the way back in exam 1. All coming full circle. So in this case, the final action is breakdown of the glycogen molecule to yield glucose, followed by its oxidation and energy production. CAMP-dependent phosphodiesterase catalyzes the destruction of the cyclic form of the CAMP, producing the non-cyclic form AMP. Thus, this phosphodiesterase enzyme or its activators can break the pathway of signal transduction and stop this hormone-induced glycogenolysis. And in each step of this cascade of reactions, the signal is amplified multiple times and thereby speed up the process. So here we have that myocyte and the hepatocyte. Myocytes will have epinephrine. Hepatocytes will have glucagon acting mostly on them. But again, same thing is sort of happening. ATP, cyclic AMP, adenylyl cyclase. We continue that activation all the way through in order to have the glycogen being broken down into the glucose 1-phosphate. And see how it expands constantly, 20 molecules. It drops a little bit to 10, but then 100, and then 1,000, and then 10,000. So it cascades. We talked about that. We talked about that when we talked about cell signaling way back when. Okay, we talked about breaking down the glycogen. Now let's actually talk about a little bit more about the synthesis of it. So glycogenesis or glycogen synthesis starts with the conversion of free glucose, so free just meaning that it's out there, it's open, to G6P catalyzed by the hexokinase or the glucokinase. Hexokinase in the muscle, glucokinase in the liver. The G6P is then converted to G1P by phosphoglucomutase. It's the same enzyme that switches them and splits them apart is going to be putting them together as well. Now, continuing to the glycogen synthesis. Once the glucose 6-phosphate is formed by the action of hexokinase or glucokinase, the next 3 enzymes are needed to synthesize glycogen. There are 3 enzymes needed to convert G1P glycogen, and they are UDP, glucose pyrophosphorylase, glycogen synthase, and glycogen branching enzyme. UDP glucose pyrophosphorylase catalyzes the reaction between glucose 1-phosphate, or G1P, and uridine triphosphate, UTP, to form UTP glucose and pyrophosphate. In the next step, UDP glucose acts as the glucose unit donor. Hydrolysis of the dual phosphorus, the pyrophosphate, provides extra exergonic energy to drive this reaction. So here we have bonds breaking. So phosphorus comes off of here, phosphorus comes off of here. The glucose and the UDP combine, and you end up with these two together. And this will break in order to release additional energy to allow for this reaction to continue to occur. So remember, when we talk about metabolism or catabolic reactions tend to break things down. Breaking things down releases the energy that was stored in chemical bonds. Glycogen synthase. The enzyme glycogen synthase catalyzes the transfer of glycosyl residue of UDP glucose to a non-reducing end of a branched glycogen molecule, making a new alpha-1,4 glycosidic linkage, and thus the glucose chain is elongated by 1 unit. So I actually misspoke a little while back about which end they build off of. They're going to continue to build on the non-reducing end. They build from this end, continue it this way, and continue to break down from this end, and break it down from this end as well. So basically the uracil, this UDP glucose, so this is the UDP part, that's the glucose, comes over here, and literally the glycogen synthase transfers the glucose onto that new end to continue to create this chain. Now the glycogen branching enzyme, alpha-1,6 linkage are formed by glycogen branching enzyme, aka amylo to transglycosylase. So 1,4 to 1,6 transglycosylase. It transfers 6 or 7 residue segments from the end of an alpha-1,4 linked glycogen chain to the C6OH group of a glycosyl residue on the same chain or another chain, and forms alpha-1,6 linked branch. So literally what it's going to do, it's going to chunk some of these out here, and it's going to bring it and reattach it. Kind of like similar to how it broke off the 3, how in glycogenolysis, the breakdown of it, it took the 3, broke it off, and moved it to the other end. This is essentially the reverse process that we see here. Sorry about that, dealing with a little bit of allergies right now. Okay, so the glucose residues can be added to the new branch further by glycogen synthase. So then they keep non-reducing end, and so then they'll just keep adding to here. It gets a little bit too long, especially like down here, it gets a little bit too long. It just breaks it again, sticks it down here, and then it keeps building. The biological significance of branching of glycogen molecule: to make it more soluble, to increase the number of non-reducing ends, increases number of sites accessible to the glycogen phosphorylase for the glycogen breakdown, as well as the glycogen synthase for glycogen synthesis. So we can break it down faster and we can build it up quicker. Body's all about being efficient, as well as saving as much space as we can. This also increases the rate of glycogen synthesis and decreases breakdown of glycogen during synthesis. We got a little bit more to go. Glycogenin primes the initial sugar residues in glycogen. So glycogen synthase cannot initiate a new glycogen chain de novo, or that's from nothing. It needs a primer. A pre-formed alpha-1,4 polyglucose chain or branch having at least 8 glucose residues. Glycogenin protein serves as both. It serves both as the primer on which new chains are assembled, as well as an enzyme that catalyzes their assembly. So glycogenin is a glycosidic bond formation between the glucose of UDP glucose and the tyrosine residue of glycogenin protein. The addition of several glucose residues, up to 8, forms a primer. So we have our glycogenin, and it's going to start the process repeating several times to start the initial chain. And then we can see here how we get these branches then forming off of this initial chain. So this glycogenin forms the primer, and then it can start to have branches and branches and branches come out of it. So glycogen synthase, that's really all you need to understand from this whole part, is that the glycogenin acts as the primer and the enzyme in order to allow glycogen to form. That's the main idea that you want to focus on with that. Glycogen synthase is regulated by covalent modification. It's phosphorylated glycogen synthase B is inactive, and dephosphorylated glycogen synthase A is active. Good thing to note here, pay really close attention to whether phosphorylation means active or inactive, and keep those straight, because earlier, if we come up, let's see if I can find, there we go. Earlier, with phosphorylase, the phosphorylated form was the active form, and the dephosphorylated is the inactive, way up here with the phosphorylase. But down here, now the dephosphorylated is the active form. So make sure you keep an eye on that and keep those two straight, because that's an easy test question that they could ask. Glycogen synthase kinase 3, GSK3. GSK3 phosphorylates glycogen synthase and inactivates it by forming phosphorylated glycogen synthase B. The primary reaction GSK3 action requires prior phosphorylation by GS by casein kinase 2 on a nearby residue. Phosphoprotein phosphatase removes phosphate group from GSB and form catalytically active GSA. Again, it's just a lot of different chemical names. Take a little bit of time to read through this. Understand what the priming reaction means. What does priming reaction mean? Know the name of this. I would also recommend knowing casein kinase 2. And really that's just know those couple of details and you should be fine. Let me make sure. Yeah. GS is glycogen synthase. GA, glycogen synthase A is the active form. Glycogen synthase B is the inactive form. Just keep that in mind. So the effects of GSK3 on GS activity. So GSK3 action requires prior phosphorylation of glycogen synthase by casein kinase 2. The activators are insulin, G6P, and glucose. Inhibitors are glucagon and epinephrine. Which, if you think, you really just need to know the one side, like what sort of things activate glycogenolysis, and you'll know that the opposites stimulate or inhibit glycogenesis. Here's all of our fun stuff. So glycogen synthase needs to be prepped with CK2, the ATP, and then GSK3 can finish phosphorylating to give finish phosphorylating to give us the inactive form. So remember, the phosphorylated form is inactive, the unphosphorylated or dephosphorylated is the active. Inactive, active. This is just going to be a lot of words here, guys. This is a repeat, literally, of the action of insulin binding and doing its thing. I'm not going to get too much into that right now, just because you really won't get asked a lot of questions on this, and it's just a lot of. I would recommend reading through this and review that insulin reaction, but mostly focus on this guy here. Hormonal regulation of glycogen metabolism. Again, this is saying the exact same thing we've heard 30 times. Insulin stimulates glycogen synthesis in liver and muscle, and the insulin increases uptake of glucose by myocytes and adipocytes. Whereas the glucagon does all of this stuff. Okay, hopefully all this hormonal regulation is just a review for you guys. Now, glycogen storage diseases. Glycogen storage diseases are a really big deal, because if we can't store glucose, then literally we can't have any energy reserves at all. So if we ever go into the fasting state, the body immediately starts to have to break down non-carbohydrate sources of energy. So there are different groups of inherited disorders characterized by the deficit of mobilization of glycogen and the deposition of abnormal forms of glycogen. So there's von Gerk's disease, which we have defective glucose 6-phosphatase. So the liver, the renal convoluted tubules are loaded with glycogen. The primary organ affected is the liver. We'll see enlarged liver, and also the kidney. We see kidney failure. Pomps disease, defective or deficiency of lysosomal alpha-1,4 and alpha-1,6 glucosidase. This is the enzyme that degrades glycogen. The primary organ affected is the skeletal muscle. You get muscle defects and muscular dystrophy. You also have cardiac muscle, you have cardiomyopathy. Forbes-Ricori disease or limit dextrinosis. You have absence of the debranching enzyme. So this enzyme causes accumulation of branched polysaccharides. So essentially you get a whole bunch of glucose, you get those branches of glucose, and you can't undo those branches. So the primary organ affected is the liver. You get an enlarged liver because, again, you can't break down the glycogen that's in it. But you also have skeletal muscle, cardiac muscle being affected. Anderson's disease or amylopectinosis. You have absence of branching enzyme. This enzyme causes accumulation of branched polysaccharides. Again, same sort of symptoms. Enlarged liver, spleen, myoglobin in urine. McArdle's disease or amylopectinosis. You have absence of muscle phosphorylase. Muscle has high levels of glycogen, exhibits diminished tolerance to exercise. This should be these should be over more. Continue with McArdle's. You get exercise-induced cramps and pain and myoglobin in the urine. And then finally some key concepts. So glucose 1-phosphate is a metabolic link between glycogenesis and glycolysis. It's the link. Excess glucose is converted to glycogen primarily in the liver and the muscle tissues when ATP supply is adequate. Glycogen is degraded to yield glucose when tissues need ATP, mainly in muscle tissue during contraction exercise, and in liver tissues to supply to other tissues via blood circulation, as well as to adjust blood glucose level. UDP glucose. The glucose unit donor in glycogen biosynthesis also donates UDP, produced here as a byproduct to feed in UDP glucuronate biosynthetic pathway, which eventually contributes to form conjugated products with drugs and other toxins and their metabolites in the detoxification pathway. UDP glucose is also used in the metabolism of galactose and in the synthesis of glycosphingolipids, which those lipids we'll talk about next try. But yeah, those are our main key concepts, and that is that whole chapter. There's a lot of technical kind of sounding words in this chapter. Focus and zoom in on the main and the big ideas. Don't spend a ton of time trying to kill yourself memorizing each of these individual names of everything. Understand the process by which things are happening, and then remember the key enzymes, kind of what those key enzymes do, what they act on, and why it is that they're necessary. And then if those key enzymes are not working, what sort of conditions or diseases do we end up with? Okay, so that's this chapter 13. And in all actuality, there was a kind of an overview. The next chapter will get more into the specifics of gluconeogenesis. Actually, pardon. We talked about glycogenesis. Now we're going to talk about gluconeogenesis. Gluconeogenesis being we do not have any carbohydrates, and so we're going to need to create glucose and create energy from non-carbohydrate sources, which we've already touched on a little bit in previous chapters. Okay, study hard and good luck.